And good afternoon, everyone. So I want to thank the folks from Adiseo for putting the symposium together and inviting me here today. We have, the task I've been given really is one, as being the first presenter. I have to keep you awake after lunch because all your blood's from your head now in your gut, right? So I suppose that's apropos for this topic this afternoon. And really the other task I've been given really is to talk a little bit about to set the stage for what we're talking about with this ecosystem of the gut and what it really means and challenges that we're facing, right? So I know we're not in the U.S., but if we look at the U.S. market right now on where we are on NAE systems, it's probably 50+% of the market broiler today being reared in an NAE system. And it's been amazing to see that story transcend over a period of about 3+ years, really from next to nothing. Few challenges along the way, but I really appreciate the companies that have put forward, and they've done a really good job of dealing with those challenges. So I always try and put my nutritionist hat on when I think about the gut, and then those of you who have seen me talk a number of times, I do kind of have that spin to it, but with a little bit of a physiological twist, because we really need to understand how that body is reacting to whatever it is faced with, right? So baseline, at least, and realize that the gut is not a static system at all. But if we look in general across critters, the gastrointestinal tract consumes roughly about 1 out of every 5 calories for maintenance, which is amazing, right? Turnover of roughly 50 to 75% per day. That changes, and we'll talk a little bit about those changes. And 1 out of every 4 proteins is actually secreted into the gut. Right? So all of that is to maintain 2 real different functions, assimilate nutrients from whatever we're eating, but protect the animal from what it might be challenged with, right? So very, very different functions, and everything's happy, it goes along. So what I'd like to, I always try and emphasize that it is not a static system. Everything is changing throughout the day, every day of the week, depending upon how that animal is reacting to its environment. Changes happen in terms of peristalsis, epithelial turnover rates, which encompasses changes to rates of proliferation, programmed cell death in the case of pathogens, actual necrotic cell death, and differentiation of those cells towards different purposes. We see changes to mucin production as well as composition, changes to barrier functions, changes to types of secretions being produced, functions of those genes, immunological responses, microbiota responses, and the list goes on and on. And we'll hear about a number of those types of changes in this ecosystem of what we call not only the bird itself, but what's happening within the context of the gut itself. So let's talk a little bit about peristalsis, really. And as we look at this, and Mike will talk about this a little bit more on the nutritional intervention side and what we are trying to deal with. But what I'm trying to convey, at least through this slide, is just really the problem that we're dealing with, right? So subclinical challenges, what we're looking at in terms of just apparent metabolizable energy. This is with a 12 times normal dose Coxivac challenge. 7 days after challenge, we see this dramatic drop from about 3,000 kcals per kilo all the way down a little bit below 2,000. An amazing detriment over a very short period of time. As far as dry matter retentions, we're looking at roughly not quite a 10% reduction in apparent dry matter retentions, and with apparent nitrogen retentions going a little bit more beyond that. So we're dealing with this, right? So this is the overt ability of the animal to assimilate nutrients and then cope with what's going on. So all of these obviously is at the cost of growth. And part of what we conjecture, at least, is can we mitigate some of these symptoms that these birds are going through to respond and deal with the challenge that's been put forward to it. And our best hope is that it deals with it quickly, sheds the pathogen, and builds some sort of longer-term response to be able to deal with it so that that period of time where that growth suppression loss of nutrient assimilation and energy assimilation is at its minimal. Just in terms of standardized ileal digestibilities, this is as a percent of diet. Just gives you an idea how many points of each amino acid loss that we're experiencing. If you look at lysine with this type of challenge, 6 days post-challenge, we're looking at about 17 points of lysine in the diet. We're looking at 4 points of methionine, and threonine, you're also looking at about 17 points, which is quite considerable. But realize this is probably towards the peak of that particular challenge as it's going through that process. Dealing with it, we'll talk a little bit about how that animal deals with it and if it's able to deal with it, just briefly as well. Well, let's first talk about some of those responses. One of those responses I always like to talk about dogma, right? So if I asked audiences typically, how long do you think it takes for a new cell in the gut to essentially replenish itself? Oftentimes we also always say 48 hours. Who thinks it's 48 hours? Nobody. Okay, you've listened to me before. Thank you. But this process is a very dynamic process, right? And it's cells going through mitotic division. Along the migration along this cryptphylus axis, they're going through a terminal differentiation, which they ultimately go through a programmed cell death and ultimately slough themselves off into the lumen of the gut. And this dynamic process of birth, life, and death is essential to that integrity of the intestinal epithelium. Now, if you remember prior, I talked about during that course of what is ailing that bird, it can have different directions along that, how many cells are going through proliferation, how that differentiation process is happening, as well as those undergoing programmed cell death. So for graduate students, I always encourage graduate students to go back into the literature. We do call it research, but hopefully you all aren't recreating the wheel. Even with some of the new toys that we have today, I want to make sure that one of our friends at University of Arkansas, Walter Bocci, I love his term, and I'm going to coin this forever, is getting lost in a data tsunami. And with some toys that we have today, it's very easy to get lost in a data tsunami and recreate some science that we had known for a very long time. Okay? If we look back at Amondi and Bird back in 1966, they were looking at this advent of trying to figure out where we were. Subtherapeutic antibiotics at that period of time were coming on the scene, and they were trying to figure out the functionality of what they did in terms of improvements in feed conversion. So they injected tritiated thymidine into the peritoneum of birds, killed birds over time to essentially see where those labeled cells had migrated along that cryptphylus axis. Fairly simple process. We do that today with varying non-radioactive types of intermediates. Well, it was interesting, you look at birds collected along 3, 8, 18, 24, and 48 hours. You can see how far those had progressed along the cryptphylus axis. And lo and behold, you look at the jejunum at roughly about 48 hours, it had traversed about 88% of the length of that cryptphylus axis. Now, that's where a lot of us have taken that dogma. We only read the abstract, and a lot of that then perpetuated literature, not only in the chicken world, but also the human world and the pig world and the mouse world, all because of that single chicken from that particular point in time. And that was only the jejunum. That was only one part of the small intestine, which is amazing because it only had gone halfway if you look at the duodenum or the ileum, right? So if you go and then calculate epithelial turnover of that system, in the jejunum, normal system, it took about 2.5 days, about 5 days each in the duodenum and jejunum, or essentially a replacement of about 20% in the upper and lower small intestine, and in the jejunum, roughly about 40% turnover per day. Now, others went on. Mary Coates, I believe she was in England, if I remember correctly, Did that in both a conventional environment and a germ-free environment to try and see how this alters in different types of environments. It was interesting, in a conventionally reared environment, that turnover here in the ileum took those birds at roughly 3.8 days, turnover about 26% per day. But in that germ-free environment, really, that was a 9-day turnover. And that's an amazing difference in magnitude of response, if you think of that from a maintenance perspective, to that intestine. So word to the wise, we always have been taking snapshots in time in terms of histology. Histology is very difficult to capture a very dynamic system. I'm going to do a Carl Parsons. Let me repeat that, please. In terms of histology, it gives us a snapshot in time and does not capture an ever-changing dynamic system. So we always have to put those in context with what else is going on. There are some things we can gather from histology, but not everything, right? Give you in terms of a coccidial challenge, 1973 paper. We've known this for a little bit of time. Emeria cervulina challenge, 250,000 sporulated oocysts, 3 and 4 days post-challenge. Light blue bars here, unchallenged birds, and you see the challenged birds having a 300% difference in turnover, about 30% turnover per day in the duodenum, but that persisted then to the jejunum and the ileum. Very dynamic process. It is very, very responsive. You can see by the graph over here, one of the things that's happening, when I talked about that life, the birth, life, and death of that villus, you can see that that tissue is responding. Obviously, it does not have the tip of the villus as you would think of it, but yet that proliferative pool is very, very different in that challenged bird. So it is trying to replenish those enterocytes very, very quickly. That turnover is happening very, very quickly. So the time at which those cells are alive is very short. But it is responding by increasing that proliferative pool. And over time, as that challenge dissipates, it will live a little bit longer and you gain back that length of the villus. Bob Teeter at Oklahoma did these studies in chambers a few years ago just to give you a snapshot of what this is in terms of the spectrum of coccidial challenge as we see it from 0 subclinical lesions to overt clinical lesions at varying ages from 20 to 48 days of age. But as that bird grows and that delay of challenge to that bird happens, because the bird is bigger, it's having to have more maintenance cost to that overall tissue. Something good to keep in mind as we see things differentially within when things are happening out in the field. Consequences of those differences in turnover rates, and this is not a bird example, but rather a pig example, gives you an idea of where the disaccharidase expression is happening along that crypt-villus axis, right? So more glucoamylase being expressed further down in that crypt-villus, that villus axis, with then a little bit more maltase coming along as you move up, starting to express more sucrase and then lactase as you move very much more towards the tip of the villus. This dynamic is important because it does take time for those cells to differentially express some of their functionalities. Jahava, I don't know if Jahava's in the room. There you are. I love this study, so I always pull it. I know it's a little old now, it's dated, it's over 20 years old, but it's classic, right? And you could explain it better than I could. Yeah. They created different pools of cells, essentially going from the crypt region on the right all the way to the villus tip. Just to look at, on the upper side, you're looking at maltase activity, and on the bottom, you're looking at sucrase activity in the chick. Relatively mature chicks, 3 to 5 weeks of age. It was very, very interesting. As you look at this, you understand that maltase activity is also expressed much more along the upper portion of that villus. Similarly, sucrase activity, more up along that upper side of the villus. So 30 to 40% more in that upper 40% of that crypt-villus axis. Now, I mention this partly because that absorptive, digestive and absorptive capability, if those cells aren't around long enough, they aren't able to express those functions. So it's interesting to see folks looking at different things now that we can measure things via gene expression. And I can tell you, if you're looking at the whole tissue and you just have not as much villus there, I can tell you that some of the things you're going to measure from a nutrient transporter, I could predict fairly easily what those are going to be, just because you have a very different mucosa to submucosa relationship. So we need to think through some of these dynamics as we're doing some of these studies to make sure that we're understanding all of those. Going to fast forward to look a little bit at mucin production. There are some interesting things that happen to this protective layer of the gut as we move into a challenge, so to speak. From a nutrient perspective, we know that threonine is 5 to 7% of that endogenous amino acid loss in the diet. Tayo, who's now at University of Kentucky, has shown that. We know that 30% of that loss of mucin, 30% of that threonine is mucin in the case of the pig. It really is that very first line of defense. And part of the question is, we know that it changes in dynamic, at least from different cereal grains and from different non-starch polysaccharides, but we know we can intervene, at least from different feed additive perspectives, to try to alter not only how much is produced, but then also potential composition of that mucin. And that is probably key, I think, in one of those unexplored areas because really that is home and interface to a lot of that microbial community. So those folks looking for a new research area out in the gut, I think that's fodder for one of those. Earlier studies we had done in diets without carvacrol and with carvacrol supplementation, we see that the carvacrol supplementation really increases total goblet cell numbers. And really that's as a particular response to what is happening within the lumen of that tract. Further studies that we had done looked at both corn, the first 3 bars on the left, and wheat-based diets, the next 3 bars on the right, with and without carvacrol or thymol. The second bar is carvacrol, the third bar is with thymol. And I think the most interesting thing that we saw with this was twofold. One, we could alter expression of the predominant secretory mucin, MUC2. We also saw that it was transcriptionally regulated by essential oils. The other thing we did, we pulled out the mucin, essentially purified it, and then essentially dried it down to essentially see how much water was within it, just to give us an idea of chemical composition of that mucin. And as we would expect, the wheat-based diets, yes, they didn't hold as much water, meaning they were more viscous, versus those, as you see in the corn-based diets on the left, a little bit. But what it also told us on that post-translational modification was that with some of these essential oils, we can alter that chemical composition such that in the face of having no enzyme present, we could change the hydration capacity of that mucin, which is really cool if you think of that as to how that dynamic is going to play out then into interface with different microbiota in the gut. It's relevant, especially because we know that perfringens is a mucolytic type of bug that likes to take advantage of some of those things as well. Well, we know that the gut is chronically dealing with these different insults and challenges. The question is always, does the animal get access to that substrate, or is it going to be the microbiota? Simply take a carbohydrate. Gross energy is 4 kcals per gram. If it gets converted to a volatile fatty acid, that only gives you 2.8 kcals per gram, relatively speaking, a 30% loss in gross energy. Huge difference. Kind of coming to the question of, Can we deal with part of these challenges? And we'll hear a lot about immunological challenges to how we're dealing with some of those in terms of the ecosystem of the gut. From Tayo's previous study, I showed you the left part of this earlier, at least on the differences on energetic utilization due to this 12x challenge of Coxivac. In terms, if we're only considering the energy loss differential, we were talking 3271 kcals in the unchallenged birds versus the 1785 in the challenged birds, a differential of nearly 1,500 kcals per kg. Now we know if we try and account for that, that's probably over an 18.5% fat diet. And I don't know anybody that could feed commercially an 18.5% fat diet. It just is not practical, right? So we know right off the bat that these energetic drains are difficult to really deal with unless you're addressing the core of that animal's response. Okay? Dealing with that pathogen, hopefully not causing too much damage, but dealing with it very quickly to get rid of that pathogen. So we developed a second follow-up study really to see if we could deal with that, at least from the terms of that amino acid uplift. And in this particular study, we did this with a full complement of lysine, methionine, threonine, isoleucine, tryptophan, isoleucine, and valine to bring back what had been lost in terms of that differential in amino acid utilization. Body weight gain, at least with this diet versus the comparative challenge group, no difference in 3-week body weights. However, we did become somewhat intermediate versus that of the unchallenged control, meaning that were able to pick up that differential in feed conversion. So can we account for this nutritionally? There's been different means and strategies to do so. Well, energetically, probably not. From an amino acid perspective, possibly, and partially, but that is only part of the story, right? I'm running short of time here. The last I'd like to touch on only just briefly is partly also a cost-based type of process that at least my brain tries to process from an innate immune perspective. We know that this response really causes a reduction in feed intake for a period of time. It causes catabolism to produce a full complement of those amino acids, which is different for those particular acute phase proteins, as well as the complement of different immunological proteins that are being produced. Sum total, we know beyond that feed intake reduction, sum total it's about 10% of nutrient use, but all of that is also variable depending upon the amount of pathogen and how virulent that pathogen is that animal is dealing with. That complement of amino acids is very different. The example I have here is just on immunoglobulins, as you see in the inset on the right. Whole body threonine content on the left bar versus mucin in the middle and IgA on the right. So very, very different in terms of one of our third most limiting amino acids. Mucin composition, as you just saw prior, very, very different versus whole body protein. So if I could sum up at least take-home messages, I want you to all realize that this is a very dynamic system, and it really is truly a system. It is adaptive to those responses to try and deal with the challenge as well as that ingredient matrix, and it will alter those endogenous maintenance, nutrient energy digestibility, as well as secretions to deal with that particular challenge. The level and how it deals with that challenge is really dependent upon the amount of pathogen, the virulence of that pathogen, and other things that would predispose that animal to a differential type of response, whether that be cocci or other things within its environment. Now the time, and really it is a time spectrum of that immunological type of response, and then recovery from that response will really vary nutrient need. And for us that are used to weighing things on a biweekly basis or a weekly basis, realize that oftentimes as we're looking at some of these strategies, we may miss some of those responses just because of how we've done other traditional types of studies. And in these cases with this very dynamic system, being able to pick up those differentials, it may not be such if you're weighing birds every 2 weeks, or if you have variance in that population from individual to individual. But it does, and because of time, change nutrient use efficiencies and changes to that tissue maintenance needs. So with that, I'd like to turn back to the moderators at this time. So thank you.